Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
New research challenges a long-standing assumption about how unicellular organisms communicate and coordinate their behavior with one another. According to the study, published in Science Advances on Wednesday, December 15, at least one species of diatom can send and receive signals that previously went undetected by scientists. Scientists assumed diatoms, which are single-celled phytoplankton, could only signal and communicate with one another by secreting infochemicals. The new study suggests that the pelagic diatom Pseudo-nitzschia delicatissima can also communicate with others through red and infrared autofluorescence triggered by exposure to sunlight. When exposed to lights of the right frequencies, diatoms in a lab synchronized their behavior, aligning vertically in the water and wobbling in time with one another, suggesting that they're capable of coordinated social behavior. To learn more, the scientists spoke with Idan Tuval, a physicist-turned-microbial ecologist at the Mediterranean Institute for Advanced Studies, about how he and his team made the discovery and where the field can go from here. The scientist: "Tell me more about your background and what led you into the field of fluorescent diatoms, of all things." Idan Tuval: "Okay, so I'm a physicist. I actually started as a theoretician long ago, and then slowly drifted into addressing questions of the world, and the interface with biology just happened naturally. I was looking at fluid mechanics, and then chaos, and mixing, and things like that, which just happened to be everywhere in biology. In particular, I slowly drifted towards the behavior of microorganisms and how physical constraints are involved in the behavior and the development and evolution of life forms. I have been, since then, trying to combine experiments and theory as much as possible. After more and more interaction with my colleagues here, first got into marine microorganisms. It came from fieldwork that some of my colleagues here have been doing. We're helping them develop a technique to address questions related to the structure of phytoplankton communities in situ. They were getting water samples at the time and, in some cases, that got into large blooms of pennate diatoms. We figured that we can use some signature out of this metadata to analyze the orientation of particles in situ and try to understand how these diatoms are oriented and distributed, not only in terms of position but in terms of orientation with respect to the vertical in the water column. So we managed to develop a technique to do that. When we applied it to some field data, it clearly showed that in many cases, these diatoms were not just randomly oriented, but instead they tended to be very clearly aligned under low shear conditions or calm waters. So how do they find each other, in particular, for things like sexual reproduction? That's a big question. We did microscopy, tracking, looking at orientation in the lab. And we published a paper on encounter rates, among the diatoms, based on this particular orientation. Looking more carefully at the data, we realized after publishing that paper that there was something more happening there. It was that they were not just sinking, but they were actually oscillating around the axis while they were sinking. So again, we wondered about the consequences of this little wobbling. We wanted to characterize it and understand if it has any influence or any consequence on the life of the community or the population that they have. There was some info in the literature about diatoms having encoded in the genome the genes for some photoreceptors in the red and infrared band. There was no clear indication of why they would have that. We're talking about organisms that live down in the water column most of the time. They live in a very bluish environment, no red light around. We just thought that maybe, we wanted to test these hypotheses. Of course, they can actually sense each other. They have chlorophyll autofluorescence. That might actually give you a sign that this stems from a signal from the fluorescence inside. So can they sense that, and does it have a consequence on how they behave? That's where the idea came from. It's telling you that there's a clear phenomenon that has not been taken into account at all, that allows cells to sense each other and react. And that's a game changer. Idan Tuval, Mediterranean Institute for Advanced Studies TS. How do you test for that response to autofluorescence? So we were using the same device in the lab, a diffractometer, with a technique that we developed before that can give us an idea of how cells are oriented in a volume. And what we did to test for the effect of red light, of the light it will emit, was to mimic that communication externally by applying to the sample the red light and oscillating the light close enough to their wobbling frequency. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.